Well, morning all. What we're going to be doing today is a lab that I was hoping we'd be able to do at the end of the year. This is one that I was super excited for us to do, which kind of combines genetics with some of the biotechnology techniques that we've been talking about in the past week. And so, what we're going to be looking at here is a particular trait which is often associated with bitter taste. This is the PTC lab, as we're going to call it. So, PTC paper is paper that's laced with a certain chemical, and it's often used as a genetic test just for fun. People who can taste the PTC have a particular type of receptor on the taste buds that allow them to taste it. It's a G coupled protein receptor, and those who cannot taste it have a different version or a different allele of that particular gene that encodes for that protein that does not allow them to taste it. And we actually will be able to figure that out not only through a tasting test, but also using PCR, restriction enzyme digest, and gel electrophoresis. So today we're going to test this on my family and we're going to see how these results go. This extraction buffer is going to allow us to do is to crack open cells. So my children, I'm going to remove this tip because I just touched my table here. My children and I and my wife are going to swab our cheek cells. And then after we've swabbed those cheek cells, we're going to put the cells into one of these small PCR tubes here. And what we're going to do is we are going to heat these samples at 95 degrees Celsius, and that's going to help us to crack open those cells and release the DNA from them. So let's go collect those samples. All right, so I have DNA from everybody in the family now, and let me grab the tubes right here to show you. So here are those tubes. Doesn't really focus that well. There we go. All right, so you can see that they appear maybe a little cloudy. That's because we've got cells in these now. So what I'm going to do is we're going to walk over here, and I'm going to show you the thermocycler we're using. This is known as a mini PCR thermocycler. You can see if you look at it from the top, we go. We've got 8 cells in here. What we're going to do is we're just going to load our samples right into there, just like so. And then we're going to close this top. Oh, probably loosen this on up a little bit. There we go. And we're just going to tighten it down just enough so that there's a little bit of pressure. The top on this thing is heated so that it keeps the liquid inside from condensing on the top of the tube, so it's going to keep everything within those wells. All right, so here is our mini PCR, as you can see. This is a really nice little setup here. We just talked about the role of this heat block here, and then, yeah, I don't know if we'll be able to see this, but there they are. Our samples are located right here in those wells. And what we're looking at here is this is the USB cord that hooks this up to my computer. And so I can run this all directly from my computer here. What we have begun is the 95 degrees Celsius heat step. You can see that the lid temperature here is at 102.6 or so. The sample temp just around 95, and what we're tracking here is that temperature over time. This 10-minute step here is going to crack open those cells, release the DNA so that we can set up our PCR. And so we'll come back once it's time to set that up. All right, so we're ready to set up the PCR reaction. For this reaction, we're going to need 3 different reagents. First, we're going to need our PCR mix, which has a nice little green colored dye here. We're going to keep this on ice until we need it because we want to keep those enzymes fresh and active. We need some primer mix here. This contains primers that are going to allow us to amplify a portion of the gene that encodes for that G protein coupled receptor, which allows us to taste or doesn't allow us to taste that PTC paper. And then we're also going to need the DNA from our extraction, which is still running right now. So we're going to set up as much of the reaction as we can. In the meantime, I'm going to start with our primer mix here. And we're going to go ahead and we're going to add 12.5 microliters of this primer mix to each of the tubes. And to prevent any sort of cross-contamination, I am going to make sure that I change my tip after each time that I use it here. All right, so there's 3. And our last sample, 4. And then we're going to go ahead and do the same thing with the PCR mix now. Ooh, these tubes are hard to open. What's nice about this PCR mix is that it has a green color in it, which lets us know that we've added all of the enzymes and the buffers that we need and the nucleotide triphosphates that we need for this PCR reaction. And this color also is going to be really helpful when we run our gel electrophoresis a little bit later as well. All right, so the last thing to add is the DNA sample. And so I'm going to go ahead and add that off camera. But once we're ready, we'll set up the thermocycler together to begin the PCR reaction. Okay, so I said that I would set this step up off screen, but I wanted to show you real quick. We have our samples here. At the bottom of them is a little bit of precipitate. We're going to avoid pipetting that into our PCR reactions. And so I'm going to go ahead and take care of this, and then we'll pick up again once it's time to set the thermocycler to drive this PCR. All right, so here I have our samples of DNA within these PCR reactions. We're going to open up that thermocycler again, just pop these right in, and then we're going to go ahead and close that up. I'm going to unscrew the top a little bit so it can snap down, and now we're just going to tighten it again just enough so that it creates a nice seal there for us. And so let's go back to our computer now. And this time we are going to run the PTC Taster Lab. And so if we go to PTC Taster Lab right here, and we go ahead and run this, and get a look at what this reaction is going to consist of. So on the bottom left-hand side over here, you can see that we are going to have an initial denaturation. This is the initial separation of the 2 DNA strands. And then we're going to go through 30 cycles of denaturation to separate the 2 strands of DNA, annealing to allow for the primers to anneal to the DNA that we want to amplify, and then 72 degrees extension. This is the actual polymerization step. And so this is where our heat-stable DNA polymerase will be able to make many copies of the DNA for us. This whole reaction is going to take probably on the order of about an hour or so, and so we're not going to sit here and watch it the whole time. I'll pick up at some point though, just so that you can get an idea of what this little instrument looks like as it's running. But I'll see you in a little bit. All right, so we have just finished our PCR. You can see on the top of this image here, it's shown us the cycles that it's gone through. There are 30 of these blips from 94 down to 58 and up to 72 degrees Celsius. And then on the bottom right-hand corner here are the number of DNA copies that have been generated if we began from one single copy of our gene. So we have on the order of about 1 trillion copies of DNA here. We're going to go ahead and we are going to set up the restriction enzyme digest now. I'm going to explain a little bit about why we're performing that experiment, and then we'll let that proceed and get ready to perform gel electrophoresis. All right, we're back. What we're going to do now is we are going to take our DNA from each of the thermocycler reactions. So here's those PCR products. We're going to go ahead and we're going to take 15 microliters of this and put them into fresh PCR tubes. So I'm going to take 15 microliters from my sample, put it into the tube number 1, and then I'm going to do the same with my wife's sample. My son Jake's sample, and then my daughter's sample. We're now going to go ahead and add restriction enzyme to each of these. The restriction enzyme that we're using is called FNU4H1. Restriction enzymes should be stored always on ice because that helps to make sure that they retain their three-dimensional conformation. We all know that heat will denature proteins, and so we want to make sure to keep it on ice as long as possible. So I'm going to set my micropipetter to 1 microliter here, and then we're going to go ahead and carefully add a microliter of this restriction enzyme to each of these reactions. Once we're done with this step, I'm going to talk a little bit about why are we even performing this restriction enzyme digest. All right, so our restriction enzyme digest is done, and in the meantime, what I've done is I've prepared for us a 2% agarose gel to separate our DNA. To do this, I took half a gram of agarose and 25 milliliters of 1X tris acetate EDTA, which is a common reagent that's used for gel electrophoresis. After mixing them together, the agarose doesn't go into solution. It actually requires heating, kind of like Jell-O does. And so that gave us this right here. And I've also included a dye into this that will fluoresce under blue light. The dye is a particular dye that is going to bind to our DNA, and so it will allow us to see those bands. So I'm going to go ahead and take this right here. We're going to pour it into our cone or our casting tray like so. All right. And so you can see here on our casting tray, we have wells. Those wells are what we're going to load our DNA into, or our PCR sample into when we begin to separate them. So we're going to let this cool for a couple of minutes, and then we'll be ready to perform the gel electrophoresis. All right, we are now ready to load our samples of the restriction enzyme digest into the agarose gel. And I'm going to do my best to try and show you how to do this. I cannot promise that this is going to be perfect. What you can see here on the right-hand side are our wells. This purple band towards the top here is our ladder. And now what I'm going to do is I'm going to put my first sample in here. We'll try to zoom in on this a little bit. What you can see is I'm taking my pipette tip, I'm putting it into that well there, just kind of, and now I'm going to slowly pipette in to get that DNA in there, and you can kind of see that green coloring. That is our sample in that well. So I'm going to go ahead and do that again. With each of these, we're loading 12 microliters of sample into each of these wells. And this takes practice. One of the things that's hard with loading gels is that you may accidentally rip the gel. It might be a little bit hard to do that with this one, though. It is a 2% gel, which means it's a little bit more stable. Also, you want to make sure that you're actually pipetting into the well and not losing that DNA sample in this process. All right, we'll go ahead and do this with our last of the 4 samples. This lane 5 here is going to be my daughter Reed. All right, so all of our samples are loaded. I want to just kind of point out a couple of things here, and we'll come back to this in a minute. The negative electrode is here, close to where the DNA is located. Positive electrode is down here. And so remember from our lecture, the DNA is going to move from negative toward the positive because that DNA has a negative charge on it. And so I'm going to get this set up. We want to put this little contraption on the top here. I need to put it on the right direction, though. There we go. And then we're just going to go ahead and turn this on. And in a few minutes, we'll take a look at what this is looking like so far. So hang on and we'll come back. All right, so this is about 3 minutes in. You can see that our DNA here has migrated out of the wells and now into the gel. We're going to go let this go for a while longer. The next time we take a look, we will look at it under the blue light so that we can see where our DNA has migrated. Hey all, so our gel is completed. You can see here we've got some beautiful separation of DNA bands.
There are a couple of lanes that seem to have two prominent bands, a couple of lanes that seem to have three prominent bands, and then our ladder there. I'm going to take a picture of this and then label it. That's going to be part of your assignment for this lab, to analyze the results here and determine what they mean. But that concludes this lab, in which we have taken DNA from human hosts, isolated it, amplified the gene that encodes for the G protein coupled receptor that allows individuals to taste PTC paper or not taste it. And then we performed restriction enzyme digest to differentiate between the two different alleles of this gene. So, again, thanks for joining in. I really hope that this at least has given you a bit of an idea of how we can use biotechnology for research purposes.